And on this chapter, we have talked about so far, this was our first chapter of living organisms. So I mentioned there were two classifications of cells, and that was prokaryotic and eukaryotic. And we talked about the characteristics of all cells, including like a cell membrane, cytoplasm, which had the cytosol and the organelles, and the plasma membrane, cell membrane, biological, that all the same thing. We also talked about them all having genetic material. That was the last thing that they all had. Then we talked about characteristics that were specific just to prokaryotes. So there was a whole slide on that. And the only thing on Earth that is prokaryotic is bacteria. So if you have an organism that you discover and you understand just by analyzing under a microscope that it is not prokaryotic, it is not a bacteria, then everything else on Earth is eukaryotic, okay? And that includes protist, fungi, plant, and animal. So then towards the end of our last lecture, we started talking about the characteristics and the organelles of eukaryotic cells. And we spoke about the cell membrane. Then we talked about the nucleus. And we talked about ribosomes. And that's where we left off. And before I get into this slide, because this is an overview slide of the structures we're about to discuss, there are three terms I want to cover with you and make sure you understand before we move on. The first term is lumen. Lumen. Lumen is, when you reference the lumen, you're talking about the opening. The opening. So when I look at this structure here, so this is the full structure, the lumen is this open space in the middle. When I talk about a blood vessel, we use the term lumen a whole lot in anatomy, but when I look at a blood vessel, the lumen is where the blood flows through. So the blood vessel, the walls of the vessel create the structure, and the lumen is that open area where the blood flows through. So the lumen is like a pathway or a passageway. Okay. So it's good times. Okay, so lumen is the first word. The second word is cisternae. And you might see it spelled cistern, or cisternae is normally the way it is referenced. But cisternae is this folding or looping that these membranes do. Like they make these folds, so we call them cisternae. So lumen is an opening, and then the cisternae is the folds. The third and final term that I want to bring up is vesicle. And it is defined on this slide. It's the only one that is defined on this slide. But vesicle. And it mentions here that it is a small membrane-bound sac that transfers substances between parts of the system. It is a small membrane-bound sac that transports substances between the parts of a system. These here are examples of vesicles. And I'll explain as I get a little bit further. But vesicles. Okay, so lumen is the opening. Cisternae are folds. And vesicles are these membrane-bound sacs that transport different substances throughout the system. The aspect that we're about to look at is called the endomembrane system. Okay, so endo tells you what? Inside. And membrane, what type of membrane am I talking about? It's a cell membrane, but what else do we call it? A plasma membrane, what else do we call it? A biological, and I'm looking for the fourth one, is the one I really want right now. Phospholipid bilayer. So endomembrane systems means that this is a system within the membrane. So we have our plasma membrane, and this is a system within it. Okay, so endomembrane system. And they themselves are going to be composed of membranes. So when we talk about a eukaryotic cell and we say that these cells have membrane-bound organelles, what type of membrane am I talking about that's around them? Their own, what type of membrane? Don't overcomplicate this, you just said it. It's a phospholipid bilayer. All of these organelles that we talk about that we say are membrane-bound, they have their own phospholipid, like that's what they're composed of when you look at it up close. We spoke on that Wednesday when we talked about the nuclear envelope being the membrane around the nucleus, and I showed you up close. It's just a phospholipid bilayer. So when you define prokaryote, you say that they lack membrane-bound organelles, and eukaryote cells have membrane-bound organelles. And what type of membrane am I talking about that they have? Phospholipid. It's a phospholipid bilayer around these organelles. All these organelles have their own little phospholipid bilayers. Okay, not all of them, but many of them. Ribosomes don't, but whatever. Okay, we'll get there. Okay, the vesicle is nothing more than a piece of the membrane pinching off and carrying whatever's in it to its next destination. And I'm going to demonstrate that when I get to exo and endocytosis, but I wanted you to kind of see all that together. At the very bottom, the third bullet lists the different membranes that we're going to discuss. Starting with the rough endoplasmic reticulum. The rough endoplasmic reticulum is abbreviated RER. And if you had to logically determine why we call it rough endoplasmic reticulum, why do you think we call it rough? It looks rough. And it looks rough because it has ribosomes attached to the surface. Does this feel rough? How do you know? How do you know what it feels like? Can you feel this? Can you tell me why you can't feel this? It's too small. Your fingers are too fat, right? We don't say it's rough because it feels rough. We say it's rough because it looks rough, right? So we call this the rough endoplasmic reticulum because it does have ribosomes attached to its surface. You can see clearly that this is labeled, it's saying cisternate. Those are those folds back and forth. You're going to see these terms, and I just want you to be comfortable with them. I don't want you to look at them and be like, I don't know what that is. So it's folding back and forth, and then it says, oh, here's the lumen. These are not just hollow folds. This is actually structuring a pathway so that whatever proteins are made by these ribosomes can be transported within that cell. Okay, so this is like an underground tunnel system where you can drop it in there and then it can go to whatever destination it needs to go to. Throw back to last time we were together. If it is a fixed ribosome, where do its proteins go? It is a fixed ribosome outside. Fixed ribosomes produce proteins that will be exported from the cell. Okay? So we know that it's associated with protein synthesis, the rough ER, because it does have ribosomes, and we know that ribosomes are the site of protein synthesis. So we have the rough endoplasmic reticulum, and then followed up by the smooth endoplasmic reticulum, named because it looks smooth, because it doesn't have ribosomes attached to it, right? Because it doesn't have ribosomes attached. And I really like this diagram because you can see the lumen and you can see what it looks like in 3 dimensions. It's clearly a pathway or a tunnel that functions for transport. But where the rough endoplasmic reticulum deals with proteins, the smooth does not. Can anybody tell me what biomolecule the smooth deals with? Lipids. It says it right there. It synthesizes lipids. What does that mean? It makes lipids. It makes fats. Can you tell me where in the cell I'm really going to need these lipids? The phospholipid bilayer, the plasma membrane, cell membrane, biological membranes. I need those lipids. Because, is there a chance they're going to be damaged? or something's going to need to be replaced? And the answer is absolutely. So when the smooth endoplasmic reticulum does work, it works for the cell. It works for the cell. So the first thing it does is it synthesizes lipids. The second thing it does, and it mentions it starts off in the liver, and I'm going to come back to that in just a moment, but it says that the smooth ER converts drugs, poisons, and toxic byproducts, so overall toxins, into substances that can be tolerated or more easily removed from the body. This smooth endoplasmic reticulum is the organelle responsible for detoxifying substances in your body. It detoxifies substances in your body. So if you take any type of medication, the reason you have to continue to take it, and you don't just take it once and you're good forever, is because the smooth endoplasmic reticulum is truly just breaking it down and filtrating it out. So if you're taking a painkiller, that painkiller you're going to have to take again in a few hours because the smooth ER is steady breaking it down and trying to get it out. Your body doesn't want all those toxins. If you drink alcohol, same exact thing. Smooth endoplasmic reticulum is responsible for breaking that alcohol down and filtering it out with the help of your liver and kidneys. Which brings me to my liver component that I said I'll come to in just a moment. Most people are pretty positive, and if you say what organ detoxifies your blood, people are like your liver. Your liver processes everything. In fact, every bit of your blood goes through your liver. So people are pretty familiar with that. If the test question says what organ is responsible for detoxification, liver is correct. But if the question says which organelle is responsible for detoxification, the answer is smooth endoplasmic reticulum. Fun fact, guess which part of your body has the highest concentration of smooth endoplasmic reticulum? The liver. Because it does the most detoxification. Okay? So throughout all of your body, and this is taking it into anatomy a little bit more, but if a certain area of your body does more work, you'll see more organelles geared towards that work in that area than you do in other areas. Your body's very good at specializing and making sure that you're effectively and efficiently utilizing your resources. Okay, Golgi complex. Golgi complex. It's not my favorite name to call it Golgi complex, but if it starts with Golgi, that's what I'm looking for. It could be Golgi apparatus, Golgi bodies, just Golgi by itself, Golgi complex. If it says Golgi, this is what I'm talking about. This is my second favorite organelle, directly behind our next organelle, which I'll get to, obviously, after this slide. But the Golgi apparatus, the first point says, it receives proteins through vesicles from the RER. What's RER? Right, rough endoplasmic reticulum. Fantastic. So it receives proteins through vesicles from their rough endoplasmic reticulum. Can you tell me where those proteins for the rough endoplasmic reticulum are on their way to? They're on their way out. Because remember, the rough endoplasmic reticulum has the fixed ribosomes, and the fixed ribosomes make proteins that will be sent out of the cell. Okay. So the proteins that are made by the rough endoplasmic reticulum are sent to the Golgi. The Golgi apparatus functions just like a post office, and it keeps everything nice and organized, which is why I like it, because I like to be nice and organized. But the Golgi begins with modifying, then sorting, and packaging the proteins for export. Let me give you an example. Go ahead, sir.
Yes, that's your question. So therefore, where do the smooth ER come into play?
Not even the same function.
I'm saying after the rough ER, I thought it was the smooth.
When you talk about the organization of the cell, the rough endoplasmic reticulum is immediately outside of the nucleus, and then it's followed up by the smooth. So people think progressively it goes through, but it doesn't. They don't even have to use the pathways. That's a really good question, and that's a common misconception. Yes, sir. But they totally don't do the same thing. So, but the smooth serves to make lipids, and the rough gets those proteins that are manufactured for export. When those proteins are sent to the Golgi apparatus, your ribosomes can make a whole lot of different proteins. Let's say you have some skin proteins, some bone proteins, and some eye proteins. All these proteins get brought to the Golgi apparatus, and the Golgi apparatus says, okay, your skin over here, skin over here, skin over here. Oh, wait. Your I over here. And puts the proteins with their groups. So it takes all the proteins that come in, and it groups them accordingly. So all the bone proteins are set in one area and sorted out. All the skin, all the eye, everything is separated. And then once they're separated, so it modified, then sorted them, then it will package all of these up and say, you are all going to the skin. Put them in a vesicle and send them out. It'll do the same for the eye. All you proteins are for the eye. Let's package you together in a vesicle, and we're going to send you out. And I'll get to vesicles in just a moment again. And then here we are, bones. All of you bones, we'll wrap you together, package you in a vesicle, and we'll send you out. Vesicles are transport vessels. Vesicles are transport vessels composed of a phospholipid bilayer. So when I memorize this, I memorize them as being like 18-wheelers or boats, something that transported goods from one place to another. So again, the rough endoplasmic reticulum sends all those proteins to the Golgi apparatus. And the Golgi apparatus divides it and says, all of the skin proteins here, all of the heart, all the bone, all the eye, gets them all together, then packages them into a vesicle and sends them off to the bone. Packages them in a vesicle, sends them off to the eye. Packages them in a vesicle and sends them off to the heart and to the skin. Just like our post office does. We all take our mail in there, the lady or man at the front scans it in, and then dumps it into a basket. That basket gets rolled to the back of the post office, and what are those people in the back doing? Sorting it. They're saying, all this is going to Houston, all this is going to San Antonio, this is going to Oklahoma. And once they're done sorting it, they put all the stuff in Houston, to Houston in a truck, and guess where they send it? To Houston. That's its vesicle, its transport vessel. Okay? And it will continue to do that until, of course, it's done with its job. I truly enjoy studying the Golgi apparatus because it is so organized and methodical in its process, but only the products that are going to leave the cell come to this post office. If it's going to stay within the cell, it doesn't have to go to the post office. Okay? It's only for export. Questions to further clarify that? Golgi. Okay, here is showing you kind of an example of the exo and endocytosis using a vesicle. First of all, let's just look at the words and analyze them using the roots already. What does exo mean? Out. And what does cyto mean? Cell. So exocytosis means it's going. On the flip side, endo means in, so into the cell. So look, I have my phospholipid bilayer. This is my vesicle. Here are whatever it is that I'm getting rid of, let's say proteins. It comes up here. Here's my vesicle. It gets closer, and as it gets closer, it begins to fuse and dump the contents. Let me explain this situation or this relationship because it became very difficult for me to visualize it. I said it's fusing with that. I don't get it. If this area right here, you guys were all the organelles in the cell, and this area right here was the cell membrane. If I tried to get in, would it let me in? No. It's going to say, hey, you don't belong here. So I'm going to have to say, hey, I'm here. I have to give Kathy these notes about cells, and the cell membrane is going to say, wait, we got to get that verified. And then Kathy's going to have to say, no, she's my teacher, let her in. So in order for me to come in, I have to be escorted. So the cell membrane, so like say I'm cell membrane and I'm pushing up against it, I'm coming in, the cell membrane will fold in and surround me. Like you see happening right here. This is me trying to come in. The cell membrane will fold in and surround me and then escort me to Kathy. And then as soon as I'm done doing my work, that vesicle will take me back. And then as it gets ready to dump me out, it will fuse. And then the vesicle is no longer a thing, and the contents are released. So it folds in, escorts, and then it will fuse back in to dump out. That's how these vesicles work. So if I'm trying to take something into the cell, it'll come in here, it'll form, you can see it in an envelope that pinches off, and then this will escort it to wherever it needs to go. If I'm trying to leave the cell, here it is. And this phospholipid bilayer will fuse and then dump that contents there. Vesicles are pretty awesome if you start to look into them. Not that I'm saying that you will, but further on in science you will. And they're really awesome if you think about them as transport vessels, because ultimately that's what they do. So I have to simplify it, but that's what they do. Is there any question or clarification needed on exo or endocytosis? Yes, ma'am.
So if it's going from one cell to another, is it in a vesicle?
Great question. If it's going from one cell to the other, it is not in a vesicle. The vesicles will stay within the cell. So it will not allow that vesicle to just leave intact. It'll dump the contents, but the vesicle itself will stay within that cell. And then the next cell will create a vesicle for it to come in. Good question. Additional questions or clarification?
So how does it move to?
How does it move to the next cell? It either goes through diffusion, through your blood, whatever it is, however it's going to get there. If it's a hormone, it's usually through diffusion, so it's just going from high to low concentrations. Good questions. This is my absolute favorite organelle. Golgi is my second favorite. This is my favorite. This is the lysosome. These lysosomes are initially formed from the Golgi. They themselves are vesicles, but they're filled with hydrolytic enzymes. Hydrolytic enzymes, and they have a low pH. So is that acidic or basic? It's acidic. So you have a vesicle full of acid. That's the lysosome. Now, it has a few jobs, but overall, it is there to make sure that everybody's doing what they're supposed to be doing. And if they're not, it's responsible for removing those aspects that aren't functioning correctly. So it mentions there that autophagy, so auto means self, autophagy removes non-functional organelles. For example, and I'm not initially trying to call anybody out, but say I'm the lysosome, and I like to be the lysosome. And I say, as I'm floating around, because they just kind of patrol the cell, I come across Dorian, and I say, Dorian, I know that you know everyone else in this class is doing exactly what they're supposed to be doing, but you're not. And as a result, you're holding us back. And I'm not okay with that. So I'm going to give you 20 seconds. I'm going to float away, and I'm going to come back. If you don't get yourself together, I'm going to kill you. And it does that. It kind of rolls off, and then it comes back in and checks in. And if when it comes back in, Dorian is doing what it's supposed to be doing, not it, it's supposed to be doing what he's supposed to be doing, then we're solid. I'm going to move on. But if not, I'm going to engulf him, and I'm filled with acid, so what's about to happen to him? He's going to completely disintegrate. He's dead. And then Jacob's like, wait, you can't do that. That's my friend. I will engulf you. Don't talk. I don't even make the threat. I just do it. Because if parts of your cells start to act up, that could impact your metabolism. That does impact your metabolism, and it can lead to diseases. So you want those lysosomes to be on point and completely non-tolerant. You either do what I tell you to do or you die. Period. And let's say that the whole class, you guys are all part of this cell, and I've already wiped off these two. I said, hey, they weren't doing what they're supposed to. Jacob was mouthing off. He knew better than that. Dorian just wasn't functioning correctly, and you guys are like, guys, you can't do that. We can't let her do this. Okay. And then I just digest myself, and I kill you all. The whole cell is gone. We are here for one purpose. We are here to maintain Jessica. We are here to maintain Dorian. We are here to maintain Taylor. If you are not on board, you are dead. I love it. I'm all about the lysosome life. Here's the standards. This is what we expect. This is where we're at. You'll either get here or die. There's not second place. Okay? So I'm a fan of lysosomes. I did this lecture one time, 2 years ago, and the next day a young lady brought me a shirt that said lysosome, and I love it. And I wear it, but only smart people get it. My husband is still I don't know why you wear that stupid shirt. Keep talking, I'll kill you. I'm kidding. I don't say that. But I do think it. I do think that. I'm like, if he only knew what a lysosome did. So it also mentions that lysosomes can be used to take care of things that accidentally get brought into the cell. And when I say accidentally, I'm talking about like viruses and bacteria that could potentially be brought on. So if a bacteria makes its way into our cells, our lysosomes are there to say, hey, wait, you don't belong. Destroy. And it functions as part of our immune system, so we're grateful for that. Okay? The only time you really get sick is when all of this is overwhelmed. Like there's too many bacteria and our lysosomes just can't handle it, and our immune system is compromised as a result. So lysosomes are my favorite non-tolerant organelle. Holistically altogether. So I have my nucleus, and then what type of endoplasmic reticulum is that? It's the rough endoplasmic reticulum clearly because it appears to be rough and has ribosomes studded on the outside. All the proteins manufactured on the rough endoplasmic reticulum will be sent out of the cell. They'll go to the Golgi apparatus or Golgi complex, but ultimately they will be sent out of the cell. And you can see little vesicles forming towards the end of those cisternae. Those vesicles will travel to the Golgi apparatus and drop off those contents. Those proteins will be modified, sorted, and packaged, and then they will be exported out of the cell. Well, they will be released. That's the main application of rough to Golgi to release through the process of exocytosis. Now, let's say that cell brings in something that could potentially be harmful. We have some lysosomes here. So it will fuse with that. Like let's say this is bacteria. It will fuse with that and destroy that bacteria. And then you'll get rid of it. You'll pee it out. You don't even know you do it. Here is showing you an organelle that was acting up. So this would be like analogous to what I was referencing for Dorian. He wasn't doing what I asked him to do. So I just engulfed him, peppered him with acids, and now he's dead. Okay? So lysosomes function in that way. They help get rid of things that don't belong, or take care of things that make their way in that have compromised the cell. Ultimately, they police, keep things in check. Questions on lysosomes? Okay, next organelle, the mitochondria. We are going to do a basic intro on this, but we spend an entire chapter on the mitochondria. So I'm going to give you some basic details, and then we'll move forward. The mitochondria, maybe in elementary or high school, you referenced it as the powerhouse of the cell. And totally cool, but that doesn't really encompass everything that you have to know at the collegiate level. I started off with the term cellular respiration. Cellular respiration is the name of the process that is carried out in the mitochondria. Cellular respiration is the name of the process that is carried out in the mitochondria. Cellular respiration is the name of the process that is carried out in the mitochondria. And that process creates ATP. So cellular respiration is how we create ATP. The next descriptor says that the mitochondria has two membranes. It's got two phospholipid bilayers, which leads us to kind of wonder what's going on here, because it's one of two organelles that do. So this mitochondria that we have within our cell has not just one plasma membrane, but it has two. Nothing else in here has two, just the mitochondria. And then upon further analysis, we see that the mitochondria has its own genome. What does that mean? It has its own genetic material. It has its own DNA, which is another quite interesting fact. Because the only thing on Earth that has genetic material is a cell. But a mitochondria is within a cell. It probably was a cell. So we have this theory called the endosymbiotic theory, and I'm going to explain it to you one time, and it'll apply to other things that I say today. What does endo mean? Inside. So we've already covered that. Does anyone know what symbiotic means, or symbiosis? It's together. It's a relationship. Whenever it's symbiotic relationships or symbiosis is a relationship between two organisms. There are different classifications of these relationships. There can be parasitism, where one benefits and one's harmed. There can be mutualism, where they both benefit. Commensalism, where one benefits and one is not affected. Okay, so you studied all that in high school. Endosymbiosis says we have a relationship between two living organisms, and one is inside the other. So then we're like, okay. We have a mitochondria that has two cell membranes, and it has its own genetic material. This theory of endosymbiosis says that the mitochondria once was alive by itself. It was a cell. What type of cell do you think the mitochondria would be? A prokaryotic or eukaryotic? Prokaryotic. It was a prokaryotic cell. We believe that the mitochondria at one point in time was a bacteria. Do you know what the first type of life on Earth is hypothesized to be? No. Prokaryotic cells. Prokaryotic cells, bacteria. But what was the atmosphere and what was the environment like initially? Was there grass? Big trees? Just the Earth's crust, a bunch of bacteria, the sun, and water. So during the day when the sun was out and we're on the surface of the Earth, what do you think the temperature is like? Super hot. And we have a lot of water. What does water do when it gets hot? It boils and then eventually evaporates. So it was hot and humid and muggy during the day, but as soon as the sun went down, it got really cold. We see this type of thing happening in the desert because there's not enough extra life there, trees and stuff, to help absorb all that heat. So we see temperature fluctuations anywhere from 20 to 40, maybe 50 or 60 degrees within one day. So it's really, really hot, and then it gets really, really cold. When you're really, really hot, how much work do you get done? Not much. And when you're really, really cold, not much. But when you're hot, you tend to start, as you're warming up, those molecules, we talked about this when we talked about atoms, they tend to start bouncing around. So what they believe is that there was a bunch of prokaryotes, some bigger than others. Like, okay, so I'm bigger than a lot of you. So the larger prokaryote, as the temperature was heating up, accidentally just engulfed a smaller one. Like, just engulfed it. And they were like, what just happened here? Alexis, I'm really sorry. I don't know what happened. She's like, no big deal. I'm just in here. How do I know that she was engulfed and not consumed? She's still alive. She's just like, what happened? We're all like, what happened? So how did she get two cell membranes? She had a cell membrane. I had a cell membrane. When I engulfed her, what process gave her another cell membrane? One we just looked at. It's a vesicle, but what was the process of bringing her in called? Endocytosis. When I engulfed her, she had her own cell membrane, and I formed a vesicle around her. So she's in here, just hanging out with her own cell membrane and my vesicle around her, and she's like, what are you doing? I'm like, I don't even know how to get you out. Just hang out for a little while. And it didn't just happen one time, because if it just happened one time, we'd probably not know about it. But it's hypothesized that it happened over and over again, that the larger bacteria continued to engulf these smaller, I don't want to say insignificant, but smaller bacteria. That is how they believe we went from prokaryotic cells to eukaryotic cells. We went from single cells to multicellular organisms that had multiple organelles through the process of endosymbiosis, where we just accidentally engulfed that other one. That makes perfect sense. This theory makes perfect sense in that she would have two cell membranes and she would have her own DNA. Why would she have her own DNA? She is her own self. Okay? She is her own self. And when you further study this, and we'll study this in that chapter, when mitochondria want to replicate, they don't go through regular mechanisms like you and I do, mitosis. They go through binary fission. So if you need more energy, your mitochondria just go through binary fission and fix that. Now, for those of you going into forensic science, mitochondria are fantastic discovery these last few years because we have nuclear DNA and then we have mitochondrial DNA. Nuclear DNA is the DNA that you are familiar with. Nuclear DNA is the DNA here. This is specific to you, but where did it come from? Your parents. It's half mom, half dad. Nuclear DNA is solid, and you can usually access it as long as that body is roughly new. Okay? But let's say you find a body that's been abandoned for 10 or 15 years and only the skeleton is left. The majority of nuclear DNA has disintegrated. However, guess what's still intact? Mitochondrial DNA. Now, the mitochondrial DNA is not the same as your nuclear DNA. This came from both your parents. Your mitochondrial DNA came straight from your mom. Only mom, because dad just gave you his DNA. Mom, her egg had all the organelles. So it was her mitochondria, it was all of her workings, her cytoplasms. Dad just added genetic material. So mitochondrial DNA matches your mom 100%. It also matches any sibling you have that shares the same mom. As long as y'all have the same mom, it's going to match 100%. So if I can't access your nuclear DNA, mitochondrial DNA, which we call mDNA, has a longer lifespan. And so you can extract that years and years later and still be able to match it to identify a body afterwards. So that was a pretty cool discovery a few years ago. Mitochondria. Questions on them. We have a full chapter, but just to intro, the mitochondria. Microbodies. Microbodies, I feel like, is a catch-all reference to any organelle that we haven't fully named yet. There's a lot of different microbodies. There's some like peroxisomes that use hydrogen peroxide to break down free radicals. There are glycosomes. It mentions those. Technically, the lysosome would actually be one of these, but we specifically recognize it in animal cells. So there's a lot of just these other organelles that have enzymes that carry out some biochemical reaction. When we say biochemical reaction, there are only two types of biochemical reactions. What are they? Metabolism, which is anabolic or catabolic, yes. So these biochemical reactions, biochemical pathways, that's what they're referencing, anabolic and catabolic. Okay? Cytoskeleton, what do you think that sounds like? Just pull in that common sense card. The skeleton of the cell. Do you remember what structure you and I don't have as animals that other cells do have? Cell wall. So when a cell has a cell wall, they do have a cytoskeleton, but it's not as noticeable. You and I, our cytoskeleton is very noticeable because we don't have a cell wall. We have a skeleton giving our shape, our cell, its shape and organizational pattern. So the cytoskeleton gives it shape and organizational pattern. And then I have here, it's very obvious in animal cells, not so obvious in cells that have cell walls. These pictures down here are out of your textbook, and they're really cool pictures. They were taken at a research institution. This is not what would happen if we took a picture of one of your cells. These have been dyed to fluoresce with the machine. But your cells have skeletons. And there are three types, or let me say this again. There are three sizes of bones within your cytoskeleton. Microfilaments, intermediate filaments, and microtubules. Large, medium, small. Large, medium, small. And I'll go over that on the next slide as well. But your cytoskeleton is the skeleton of your cell, and it's most prominent or noticeable in animal cells. Here, excuse me, again, these diagrams came straight out of your text. But I've broken this up. Here's large, medium, small, as far as the components of the cytoskeleton, or the bones, if you will. And so I have the microtubules, and then I have in parentheses tubulin. Tubulin is the name of the protein found in microtubules. You have to know that. So that's why I grouped these together. Intermediate fibers, which are a middle size, have the protein, they're called intermediate proteins. So intermediate and intermediate, that's pretty easy to memorize. The microfilaments, which are the smallest, are composed of the protein actin. Actin is the protein found in microfilaments. Actin is the protein found in microfilaments. So you have each of the classifications of the components of the cytoskeleton, so large, medium, and small. And then the protein associated with them. This is the large one, the microtubule. Do you remember what this open area is called? The lumen. You're going to see this come up over and over again. The lumen is the largest, and the microtubules, because they're the biggest, and it gets smaller and smaller to where it's non-existent when you get to the smallest one. So here are the three components. You and I are just going to focus on the large aspect, the large component, the microtubules. So a component of the cytoskeleton, the next slide is going to talk about specifically what the microtubules are responsible for, the largest component of the cytoskeleton. They're responsible for a lot, but there's a few things that I want to point out. This structure here 100% matches this function. It says organelle movement by motor proteins. And it also says that the microtubules provide tracks for mobile organelles. So I needed something to help me picture this. I don't know how many of you actually go to the bank, because I know that you can do all that electronic picture taking and stuff. But if you've been to the bank, you know how you put that little tube into the, I don't know what it's called. The little, the vacuum chute thing. So you put that in there, and then it's like, and it takes it to inside the bank, and the bank puts your money or takes your money out, and then it brings it back. That's literally how a microtubule works. The microtubule has this lumen. It's open like this. And if I'm on this side of the cell and I need to get to this side of the cell, I just can't go straight through. It's just not an option. So I had to find a pathway to get around. So I can jump into a microtubule, and it kind of works as one of those vacuum chutes. It'll kind of slingshot me around to my destination. And when I need to go back or I need to go somewhere else, I can get on a different microtubule. It's a cytoskeleton, so it goes all the way around. And it'll allow me to travel within that cell. It provides tracks for mobile organelles. Okay? Next thing. Besides our skeleton offering us physical support and shape, there's another function that our skeleton carries out. What does it allow us to do? Move. Our skeleton allows us to move. Cytoskeletons allow large cells to move. Now, do they move as pretty as we do? The answer is absolutely not. I can't wait to teach you about amoeboid motion, but it's disgusting and cool all at the same time. Okay? So it does allow the cells to move, just like our skeleton does. And then here it has centrioles brought up twice. So I talked about the first, that the microtubules are used for transportation within the cell, that it helps the cell move. And the last function is that of the centrioles, which I have here, of the centrioles. And the centrioles, first of all, are only found in animal cells. That's a test question. Centrioles are only found in animal cells, so you'll only find it in this cell. And the centrioles are surrounded by an area called the centrosome. It's up there. Centrioles surrounded by centrosome, and this directs cell division in animal cells. This structure directs cell division in animal cells. So when this cell goes to reproduce. That cell cycle, mitosis, is directed by the centrioles. So centrioles are microtubules, composed of microtubules. They are surrounded by an area called the centrosome, and they direct cell division. They direct cell division. Okay? On this slide right here, it shows the centrioles, and then it shows a photomicrograph, and it says a 9-3 configuration. When you look at this and its organization, it's showing you that it has 3 microtubules here, 3 here, and so what do you think the 9-3 configuration means? There's 9 groups of 3. So there are 9 groups of 3 microtubules together that create each centriole. 9-3 configuration means that there's 9 groups of 3 microtubules that create each of those centrioles. Still talking about microtubules, but now furthering their function as flagella and cilia. Many of you have heard these terms before, but the thing is that in your head, the tail is just kind of tacked on. Just like in organisms like you and I, the tail has to grow and extend from inside. So the cytoskeleton will extend into a flagella, if that cell is supposed to have a flagella. Or the cytoskeleton will extend into cilia, if that cell is to have cilia. The difference, flagella allows you to move long distances. Cilia is for local movements, very close movements. Flagella is for long distance. Cilia is for local movements. Cilia cannot just randomly move, because what direction would that cell go? Nowhere, because it would have some going to the right, some going to the left. Cilia move in a very synchronized manner, so that if things need to move in one direction, all the cilia go in that way, okay, to help that cell move. So cilia is used for very local movement in an area. Flagella is used for long distance movement. So that's mentioned there. It says here that in flagella and cilia, the structures are 9-2 configuration. What do you think that means? 9 groups of 2. So on the centrioles, it was a 9-3 configuration. For the cilia and flagella, it's 9 groups of 2 microtubules each. These terms are brought up in the text right now, but they have not been defined to date. So I want to make sure that you understand them. The statement is that prokaryotes have analogous, not homologous, flagella and cilia. Analogous means that you have the same function but a different structure. You have the same function but a different structure. You have the same function but a different structure. That's analogous. Same function but different structure. An example would be bird wings and butterfly wings. They're both used for flying, but are they made the same? No. So they have the same function, but a different structure. That's what analogous means. Homologous is the opposite. It has the same structure, but a different function. Homologous is I have the same structure, but a different function. Same structure, different function. That's homologous. Example of homologous. The bones in our arm include the humerus, the ulna, the radius, carpals, metacarpals, and phalanges. We have all those bones, and we use them to do various activities. All of these bones are also found in the front flippers of a whale. Exact same bones. But do they use them the same way we use our arms? No. Same structure. We have the same bones. We just use them differently. So homologous means same structure, different function. Analogous means same function, different structure. Everybody clear on that? So on this statement where it says prokaryotes have analogous flagella and cilia, that means that they have the same function, just a different structure. And this shows you the difference up close. It shows you that it's an extension of the microtubules. Here's my 9 sets of 2 for flagella and cilia. These centrioles are 9 sets of 3. And here's showing you the difference in the movement. So flagella moves, and that it's called a locomotive move. So it helps with locomotion or movement. And you can see kind of how it oscillates. And here the cilia are moving back and forth in a very synchronous, all-together way. Okay, organelles that are only found in plant cells. Organelles that are only found in plant cells. There are 3. The first is chloroplast. Main takeaways from chloroplast is that they are the site of photosynthesis. Photosynthesis takes place in the chloroplast. We also find that this structure has 2 cell membranes and its own DNA. Can you tell me probably why? It was once an organism on its own, but it's inside of a cell now through the process of endosymbiosis. It's endocytosis, but the theory is endosymbiosis. Endosymbiosis. And all of that is here, and it's even on the notes on eCampus as well. They carry out photosynthesis. It has 2 membranes, an inner and an outer membrane, and it has undergone endosymbiosis, which is telling us it has its own genetic material. So chloroplast. Does anybody else have an idea what the other organelles are that are only found in plants? A large central vacuole, and there's one more that we've already discussed. Cell wall. Yes. So the chloroplast, the vacuole, and the cell wall are characteristics that are unique to plant cells. So with chloroplast, vacuole, the vacuole is this large central area. What is stored there? Mostly water, but if that organism makes a sap or anything like that, it could also be stored there. The term tonoplast, here at the bottom, says it's the membrane that surrounds that vacuole. It is not depicted here to where you can see it, but there is a lipid bilayer that surrounds this, and that lipid bilayer is called the tonoplast. So the lipid bilayer that surrounds the vacuole is called the tonoplast. And the last term that I need you to know on this slide is turgor pressure. Turgor pressure basically tells us how much water plant cells have. If their vacuole is full, that plant cell stands up straight and it looks nice and healthy. It means it's adequately watered. So if you have a plant that's adequately watered, how does it look? Looks good. It's standing up straight. Everything looks happy. What about if it's a plant at my house that has not been watered? We don't call it drooping. What do we call that? Wilting. Call it wilting. The plant is able to wilt when this loses water. Because this is what kind of reinforces it and holds it tight. Like if your stomach is full at Thanksgiving, you're like, oh. That's that vacuole being filled. But as it gets empty, as you start to burn it away or that water starts to lose, that cell starts to shrivel up and it allows the plant to wilt. That's its way of saying that it needs to water. But see, I don't look at it. I'm listening. And since it doesn't talk, it dies. But turgor pressure is the name of that. The name of that characteristic where it fills that vacuole and that cell looks nice and healthy. The last characteristic of only plant cells is the cell wall. And you've already covered the basic characteristics of the cell wall. Do you remember what carbohydrate is found in plant cell walls? No, it is a starch. Cellulose. Cellulose. Cellulose, cellulose, cellulose. Yes. And that's here. Okay? It says that there's two types of cell walls, primary and secondary. What do you think the difference is? One's first and one's second, yes. So what this is trying to depict, and I want to explain it because I think if I just put these pictures up here, I'm like, oh, they'll figure it out. But it's a difficult diagram to figure out. So this is supposed to be two separate cells right here. And because they're plant cells don't move around. They're like stuck together. So we have each of the cells, then we have their cell membrane first, so their plasma membrane. Then we have their secondary cell wall, their primary cell wall, and then this structure right here is the divider between the two cells. We call it the middle lamella. So it is dividing adjacent cells. Okay, it's holding them together, but it's keeping them divided. So if we're going from the outside in, here's the division point. We go primary, secondary, plasma membrane, cell. Okay, so the types of cell walls. The middle lamella there, and all of this is labeled if you go back and study it. The middle lamella there is separating those two. The last characteristic of the cell wall that I want to point out is what we call plasmodesmata. Plasmodesmata are little tunnels present between the cells so that whatever in this cell can travel to that cell. This is really important for plants because where do plants bring in water? The roots. Contrary to what all of the people in your neighborhood, when they water the leaves don't take in the water. The water is taken in from the roots. So how does it make its way to the top of the plant? We say it sucks it up. It's a science behind it. It's called capillary action through the process of transpiration. But literally the water moves from cell to cell through these gaps called plasmodesmata. So because the plant cells can't just throw things back and forth and they can't move like our cells do, that water has to go through. So does it happen fast? No. It happens pretty slow, but it does happen. And that water can move from one cell to the other through those pores, plasmodesmata. Okay? Almost finished. Cell junctions. This is just for animals. Animal cells are held together, not by their cell wall because they don't have a cell wall, but they have two types of relationships when they're held with other cells. They can be not permanent and permanent. What do you think the difference is? Yeah, one stays and one can hold on to it and then let go. Okay? When we hold on to it and let go, it might just be for a short time. I just need to help you get to a destination and then let you go. Okay, like blood cells or something like that. But when we talk about permanent cell junctions, we're talking about junctions in between like your cardiomyocytes, your heart cells, or your neurons, stuff like that. Cells that aren't going to move. So when we talk about these permanent cell junctions, there are three types. So I have types of permanent ones: anchoring, tight, and gap. And we're going to pull that common sense card again because you know I love to pull common sense. Let's go with a tight junction. If you had to guess, what would be a characteristic of a tight junction? It would be very tight. And as a result of being very tight, nothing can really get through. So if a tight junction is very tight and nothing can get through, what do you think a characteristic of a gap junction would be? There'd be a gap and things can get through. Okay, good, good. Now we know tight and gap. What about an anchoring junction? If I was charged with anchoring, what would my job be? I'm just holding you in place. When I used the example the other day of your skin cells, and I said when you get sunburned and they kind of hold together, it's because they're stuck together. They create a tissue. It's anchoring junctions that holds your skin cells together. Your brain cells are held together by tight junctions, and your cardiomyocytes, which are your heart cells, are held together by gap junctions. Those are just a couple of examples. So the next slide shows you what they physically look like. I have my group of animal cells. And here, anchoring junctions, it's just telling you, it's just holding them in place. So here's the cell membrane of one cell, here's the cell membrane of the other, so you have two separate ones. They're just being held together. It's references like Velcro or spot welding, where you just kind of put it there and you just hold it. A tight junction, whenever I first started studying this, it was easy for me to remember because when I saw it, it looked like there was the pattern of a sewing machine. So I said, oh, well, that looks like it's really tight because it's sewn together. Not much is getting through here. This is what holds your brain cells, your neurons, and such together. And then here we have gap junctions. Gap junctions are called gap junctions because they provide a gap for things to move through. So, for example, in our heart cells, our cardiomyocytes, calcium levels have to be high in your body at all the time. All the time. Calcium and sodium and potassium. And those electrolytes will move back and forth throughout your cardiomyocytes to make sure that your heart has enough power and electrolytes to contract. And they move through these gap junctions. So, just some application there to help that make sense. Okay? Let me show you this picture first. Do you remember what characteristic you and I don't have as animal cells? We don't have a cell wall. We are the only cells that don't have a cell wall. As a result, so here's our phospholipid bilayer. Here's the proteins that I had kind of mentioned the other day that are stuck in there. Here's our cytoskeleton. So this is all inside our cell. You and I have what we call an extracellular matrix. Extracellular is like, it made me think of extraterrestrial. So it wasn't on Earth, it was outside of Earth. So the extracellular matrix is not in the cell, it's outside the cell. And it helps to reinforce and provide protection for that cell. There are a lot of different components that can be found here. And this matrix is just a textbook picture. But the extracellular matrix found in your blood cells is way different than the matrix found in your bone cells. Because your blood is liquid and your bone is solid. They're both made of animal cells, but their surrounding protection is different because of their function. So this is just kind of a textbook. It has some proteins, it has some collagen fibers, and so on. So that's what that is. Here's our last slide. The extracellular matrix. It surrounds these cells and provides structural support as well as helps the cells stick together. It exists outside of that plasma membrane. It exists outside of the plasma membrane. Then on the right side, I have some various components. These are not the only components, but these are a few of the components of the extracellular matrix. So collagen, it provides durability and elasticity. Not that you need to know all that, but proteoglycans. Proteo tells you it's protein, and glycan tells you that it's sugar, which means it's a carbohydrate. Very good. So proteins and carbohydrates put together, those usually help link or hold something or maybe change the consistency of a fluid. Take it from being very, very viscous to being less viscous. It can manipulate the viscosity. Fibronectins have a fibrous base, so those are usually pretty tight. It's telling you that they're connecting cells via integrins. These are different proteins that are integrated into the cell membrane, holding them together. You don't need to necessarily know the functions of each of those, just that they're components of the extracellular matrix. And this brings us to the end of chapter 5. Any questions? Okay, so what needs to happen?